चुका है, 1 घंटे में हमने बहुत कुछ रिवाइज कर लिया, वी हैव रिवाइज्ड अ लॉट गाइस, है ना, मजा आ गया। आगे बढ़ते हैं सर। Let us go towards the next topic right now, molecular biology. In molecular biology, yes, what is attached to the 3' end of mRNA after transcription? We will come back to this question later on, but the topic that you have to learn right now is post-transcriptional modification. Post-transcriptional modification. Welcome, 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 my dear friend. Now actually what is going to happen here, there will be first of all a DNA. That double-stranded DNA is the one which is going to form single-stranded hnRNA, heteronuclear RNA, and this is known as transcription. Okay. After that, this hnRNA will be modified to form your mRNA, and that is the reason why it is known as the post-transcriptional modification. Okay, so first from DNA we are going to form hnRNA, and that is nothing but transcription. hnRNA will be modified to form mRNA, and that is post-transcriptional modification. After transcription, that is nothing but the modification, and these two is actually taking place inside the nucleus only. After that, this mRNA will come out of the nucleus into cytoplasm. In cytoplasm, mRNA is the one which is helping in the formation of protein. By which process? By the process of translation. mRNA helps in the formation of protein by the process of translation, guys. Okay. Acha, this was asked in 2020 December question. Very good, Dr. Rajat. Okay. So I repeat again here, what is going to happen, sir? DNA to hnRNA, transcription. hnRNA modified, post-transcriptional modification. mRNA to protein, translation. And after the formation of protein, this protein will undergo again modification, and that is known as the post-translational modification. Post-translational modification, and this is the entire overview actually. ये ना complete overview है. तो DNA से पहले hnRNA to mRNA form. mRNA is going to come out of the nucleus, and after coming out of the nucleus, mRNA to protein, and protein will be modified, sir. That is about the post-transcriptional modification. Where it comes, exactly I'm telling you that one, sir. So what is the meaning of this post-transcriptional modification here, sir? We have to modify the hnRNA. There are only and only 3 points that you have to learn here. Post-transcriptional modification, there are 3 important modifications that you have to learn. And I don't know why it will be repeatedly asked in your MCA exam. So please yaad rakhiye sir. Abhi ke abhi. Everyone along with me right now. No distractions, sir. Come on, learn along with me. Imagine this one will be your hnRNA, heteronuclear RNA. This is 5' end, this is 3' end, and we all know RNA will be just a single stranded. Okay. One modification will be on the head, 5', one modification will be on the tail, that is your 3', and another modification will be in the middle, that is your body. Now my dear friend, welcome. What is a modification at 5' end? At 5' end, you remember like head, and on head what are you going to wear? Yes, cap. और इसीलिए हम उसको कहते हैं 5' capping. So what is the modification taking place at 5' end, sir? 5' end, it is 5' capping. And 5' capping, my dear friends, what will be attached here? Yes, it will be 5-methyl guanosine. You have to remember the name of this one, sir. This was the one which was asked in your MCA exam. ये आपके MCA exam में पूछा गया था. Okay. Sir, no one can teach biochem like you. My dad is also a doctor and he is watching. We are watching from UK. Amazing. Thank you, thank you, thank you for appreciating my work. My hard work is paid off. Thank you, thank you, thank you. Welcome guys. So what is the meaning of 5' capping here? Capping का मतलब यहाँ पर क्या होता है? There will be a substance attached there. What is the name of the substance? 5-methyl guanosine will be attached over there, sir. You have to remember this one. It was asked in your MCA exam. Lovely. And what about the 3' end, sir? 3' end, इसको हम कैसे याद रखेंगे? How to remember this one here, guys? Remember 3' end is like a tail, and here we are going to attach a long tail of adenine, 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 adenine. तो आप ना इसको ऐसे याद रख लो. Remember here like a tail. So many adenine there. Many means what, sir? Poly, and adenine A. There is a tail. Poly A tail. Modification number 2. So in post-transcriptional modification, modification number 2 is nothing but the poly A tail, sir. Poly A tail. Now welcome to the middle part here. In the middle part, there will be a small parts here which are known as intron, introns and all, and in the middle there will be like exons and all, sir. But you remember here, out of these two, intron and exon, introns are the one which are non-functional. Introns are non-functional. So just cut and remove the intron, cut and remove the intron, cut and remove the intron, and then ligate the exons. Exons को क्या करोगे आप? Ligate कर दोगे. And this is actually known as splicing. What is this modification number 3 known as, guys? Splicing. So what is the meaning of splicing here? Splicing means removal of introns and ligation of exons. Removal of introns and ligation of exons is nothing but splicing. That is the modification number 3. Is it okay? Now, sir, is it done automatically? No. Anything cannot happen automatically here like that. It is done with the help of certain enzyme. Which enzyme here, sir? Spliceosomes. Yes, splicing is done by which enzyme? Spliceosome. It is also known as SNRNPs. What is that one, sir? SNRNPs. Small nuclear ribonucleoprotein, sir. Small nuclear ribonucleoprotein. Small nuclear ribonucleoprotein. And SNRNPs are nothing but your SNRPs. इसी को हम क्या कहते हैं, sir? SNRPs कहते हैं. ठीक है जी? Perfect. So spliceosomes or SNRNPs or SNRPs, these are the one which are actually going to do what, sir? Splicing. And what is the meaning of splicing? Splicing means a removal of intron and then the ligation of exon here, sir. Okay. Now here I just want to make, yes. I have seen just now your messages there. Thank you for helping me. So it is 1 second here. I'm extremely sorry. Just do a small correction there. It is 7-methylguanosine. Okay, yes, it is 7-methylguanosine. Thank you all for helping me. Okay, so just remember, my dear friend, extremely important topic for your MCA exam. MCA exam में बहुत extremely important topic है ये, post transcriptional modification. How many modifications you have to remember? 3. One on the head, one on the tail, and one in the middle there, guys. So number 1, remember, phi capping. And what is added in the phi capping there, sir? 7-methylguanosine. 7-methylguanosine. I repeat again, 7-methylguanosine. And number 2 will be poly-A tail. Poly-A tail, that is at 3' end. And number 3 modification will be splicing. And splicing is nothing but done by spliceosomes or SNRNPs or SNRPs, my dear friends. Lovely. Out of these 3, definitely they'll ask you one question, answer that one. And yes, welcome back to this question here. What is attached to the 3' end of the mRNA? It is poly-A tail. Poly-A tail will be attached to the 3' end of the mRNA, guys. Awesome. Thank you. Next one, sir. Now after this one, the next thing that you have to learn in molecular biology, protein folding. Another important thing for your MCI examination. Protein folding is done by what? See, actually what is happening here, from DNA, you will be getting what? mRNA. From mRNA, what are you getting there, sir? Protein. Ab is protein ka kya hoga? What is the fate of the protein? There are 2 things. If it is defective, what is going to happen? If there is no defect, what is going to happen? If there is no defect in the protein, or if there is a defect in the protein, what is going to happen? If there is no defect in the protein, what we are going to do? We are going to do the protein folding, sir. Packing will be done. And that protein folding is done by chaperones. Is it okay, my dear doctors? Chaperones. Understanding, my dear friends? So it's protein folding is done by chaperones. Yes, in India here, we're all chaperone. It's not chaperone, it's actually chaperones. Is it okay? So protein folding is done by chaperone. Sir, if there's a defect to do, we are actually going to break down that defective protein. How? With the help of 2 substances there. One is ubiquitin along with proteasomes. Ubiquitin and proteasomes, they are the one which are going to break down the defective protein there, guys. They are going to break down the same question asked in the IMAT 2020 also, it seems. Wow, lovely. Lovely, awesome, guys. Lovely. So remember, protein folding is done by what, guys? Chaperones. Whereas if it is defective protein, that we are going to break that with the help of ubiquitin and the proteasomes. Ubiquitin and proteasomes are going to break down that once. Next one. All are true regarding the genetic code except what? All of these are true regarding the genetic code except what? Now, my dear friends, welcome. First of all, what is the another name of the genetic code? Genetic code, it is nothing but codon. And every one of you knows about the codon. I hope you have heard about that AUG, UAA, UAG and all those things. Hai na? Genetic code of the codon. Now remember, there are 4 important characteristic properties of codons. there are 4 characteristic properties of codon. Number 1, a codon is degenerate. अरे यार, ये मेरे पास तो यहाँ पर परफेक्टली चल रहा है, तुम लोग यहाँ पर बार बार इसको लैगिंग क्यों कह रहे हो? व्हाट इज हैपनिंग? Everything is like perfect from my end. Now, okay, no lagging, sir. Thank you, Dr. Arshit. Now, welcome to genetic code or the codon here, sir. Genetic code or the codon here. चलिए देखते हैं इसके बारे में. Now, my dear doctors, remember, genetic code of the codon has got 4 important features. What are the 4 important features? It is degenerate. What is the meaning of degenerate? Remember, there are totally 64 codons. But there are how many amino acids? Only 20 amino acid. So if you see opposite of this, each and every amino acid can be coded by more than one codon. That is degenerate. Yes, correct. It is universal. Yes, a codon is universal. The same codon will code for same amino acid in every species. Commaless, and remember one more thing, sir. So these are all the important 4 features of codon. So in this question, we have actually in this question we have actually covered about all these codons here, sir, and their important properties here. Okay. So there are totally 4 important properties of the codon. A codon is degenerate and it is universal. It is unambiguous, which is also known as specific here, guys. And then it is commaless and non-overlapping. Okay. Okay. So a codon will be actually commaless and non-overlapping. All these are the 4 important, sir. 4 important points here, guys. Okay. And it is playing absolutely normal here by, I don't know what is happening with you people there. चलिए आगे बढ़ते हैं, sir. Let's go ahead right now. Now moving ahead, yes, repeat the codon wala part. Okay, Dr. Raj, I'm going to repeat that one. I'm going to repeat this codon wala part here. Okay, Dr. Raj, no problem. First of all, remember what is another name of the codon? A codon is also known as genetic code here, sir. A codon is also known as the genetic code. Okay, thank you all, guys. Thank you all for genetic code. Okay, thank you all, guys. Thank you all for cooperating with me. I don't know what is happening here. First of all, remember that a codon, it is also known as a genetic code, guys. Genetic code. And remember, there are 4 important properties of codon. A codon will be actually degenerate. What is the meaning of degenerate, guys? Yes, remember, help of numbers here. Numbers से आप ना बड़ा आसान याद रख सकते हो. How many codons are there? 64 codons. How many amino acids are there? There are only 20 amino acids. So think like this. One amino acid can more than one codon. Think like this. One amino acid can be coded by more than one codon. Okay, so that is nothing but degenerate. Next, number 2 is universal. What is universal, guys? AUG amino codon. AUG will always code for methionine only in all species. In all species, not only like human beings like you and me, every species, guys, it is the one coding for methionine only. So it is universal. Yes, a codon will be unambiguous and specific, and remember it will be non-overlapping and commaless, guys. These are the 4 important properties of codon you got to remember here. Next one. Now, this is a question which was actually given in your December 2019 MCA exam. In December 2019 MCA exam, this type of question was given in the exam, guys. Okay. Now, if the content of adenine is 15%, what is the amount of guanine in the DNA? Now, remember, in the DNA, we are going to follow, according to Chargaff's rule, it says that adenine loves to bind with thymine and guanine loves to bind with cytosine with a triple bond. According to Chargaff's rule, come on, guys. According to Chargaff's rule, adenine loves to bind with thymine and guanine loves to bind with cytosine there, guys. How much is adenine given in the question there? 15%. Therefore, thymine has to be 15 only. 15 plus 15 will be totally 30%. What about the remaining here? So the remaining will be 70% here. If it is 70, cytosine will be 35 and guanine will be 35% here. And what is asked in the exam here? In the exam, the question was regarding guanine. So how much is guanine here, sir? 35% is the correct answer here. Perfectly right. I hope you got the concept here, guys. No need of any formula, no need of any heavy mathematics or something here, guys. Nothing, कोई ज़रूरत नहीं है यहाँ पर. Apply my simple logic here. तो चार्गस भाई साहब ने हमको क्या कहा था? What did Chargas rule says us? Adenine loves to bind with thymine and guanine loves to bind with the cytosine. ATGC. ATGC, sir. Adenine loves to bind with thymine and guanine loves to bind with cytosine. With the help of this simple thing, you'll be able to do the mathematics here, sir. Class done. Next one now. Now let's move on to the next important topic, that is nucleotide metabolism. Nucleotide metabolism. Guys, are you all okay? Are you all okay here, my dear friends? Just confirm me. Everyone give me a thumbs up. Everyone give me a thumbs up here. Are you all comfortable? Shall we continue or shall we stop? Is this session useful here? I hope I'm not I hope I'm actually not at all wasting your time, guys. Why? Because it's very high time here right now. We are just left out with 3 days in the exam and I don't want to waste your time actually. Is this useful to you people? Then we'll continue. Otherwise we'll stop here. यहाँ तक काफी है? Everything is perfect? Yes sir, very good. How is the Josh now? 100%. Very nice, very good sir, very good. I'm really happy. I believe you people, guys. I trust you people more than you people. You are going to definitely clear this exam. Hardly it's a matter of 150 marks, man. Out of 300, 150. It's just asking you for half, 50% performance, and definitely you are going to clear the exam, sir. चलिए quickly आगे बढ़ते हैं, sir. Let's go ahead now, quickly right now, sir. Nucleotide metabolism. In the nucleotide metabolism, the first thing that you have to learn here is about the enzyme deficient in the given image. In the given image, first of all, you can see all these fingers are actually damaged here and these lips are actually damaged here. So this is actually explaining you about the self-mutilation here, sir. This is explaining you about what, sir? Self-mutilation, harming one own self. The person is harming himself. And I told you this many times very clearly in the exam, in regular classes and even in the YouTube sessions and all, guys. Yes, self-mutilation, mostly what is the reason here, guys? It is explaining you which disease here. It is Lesch-Nyhan syndrome. It is Lesch-Nyhan syndrome. Nyhan syndrome, what is the enzyme deficiency in this one? The enzyme deficient over here will be HGPRT, hypoxanthine guanine phosphoribosyl transferase. Hypoxanthine guanine phosphoribosyl transferase here, sir. Okay. Enzyme deficiency here will be HGPRT, hypoxanthine guanine phosphoribosyl transferase. Then what will be the clinical features here? The clinical feature number 1 here will be neurological disorder. Be neurological disorders here, guys. Okay. Then next is number 2, there will be gout here. The gout, person will be suffering with gout, guys. And number 3, there will be self-mutilation. And the important keyword here, the important keyword here is actually the self-mutilation, guys. So self-mutilation, harming one own self. The person will be harming one own self, like, you know, literally chewing the fingers and things. Okay, that is what is happening in the Lesch-Nyhan syndrome. So in the given image, what is the enzyme deficient? It is HGPRT, hypoxanthine guanine phosphoribosyl transferase. Hypoxanthine guanine phosphoribosyl transferase. Okay. So for you people, all the image-based question as well as clinical questions I'm trying to include here, guys. Try to remember that. Okay. Thank you, Dr. Ankit. चलिए अब आगे बढ़ते हैं, sir. Next one. Oh my God, extremely important concept here, sir. Extremely very much important concept here. I hope you all remember the purine, sir. Adenosine monophosphate, guanosine monophosphate, and then inosine monophosphate here, sir. AMP, GMP, and IMP, guys. And all these purines, your AMP, GMP, and IMP, when they undergo degradation, they are finally going to form hypoxanthine. And from hypoxanthine, it is going to get converted to xanthine. And from xanthine, it is uric acid. Oh my God. My dear friend, first of all, print this in your mind. This is the end product. Your mind. Get it printed in your mind that what is the end product of degradation of purine? What is the end product of degradation of purine? It is uric acid. And I don't know why our super talented FMGs, they will be writing again. I request all of you people with the folded hands here, what is the end product of degradation of purine? The end product of degradation of purines will be uric acid. Uric acid, not urea, guys. Okay. The end product of degradation of purine will be uric acid. Uric acid, uric acid. End product of degradation of purines will be uric acid. Remember that. And after that, my dear friends, remember here, what is the name of the enzyme here? It is xanthine oxidase. It is xanthine oxidase here. In some people, what is going to happen? Uric acid level will increase. And increase in the uric acid level, this condition is nothing but gout. What is gout? Gout is nothing but hyperuricemia. What is gout, sir? Gout is nothing but hyperuricemia. Increased in the uric acid level. Increase in the uric acid level. अब treatment के लिए हम क्या करेंगे? We are going to give the drug as allopurinol. And what is the mechanism of action of allopurinol? Allopurinol is going to inhibit this enzyme xanthine oxidase. Allopurinol is going to inhibit the enzyme xanthine oxidase, my dear friends. Is it okay? So what is the drug used for the treatment of the given condition? Inhibits which enzyme? Oh my God. So for gout, we will be using allopurinol. Allopurinol is going to inhibit xanthine oxidase. Allopurinol inhibits xanthine oxidase. But remember, that is a drug which is used for the treatment of chronic gout, sir. Chronic gout, not the acute one. Can anyone tell me in the comment section there, what is the drug of choice for acute gout, for the treatment of acute gout? What is the drug of choice for treatment of acute gout, sir? Come on, come on, come on, come on, come on. Very good, lovely, lovely. Yes, Dr. Himanshi, very nice. For acute gout, we'll be using NSAIDs, non-steroidal anti-inflammatory drugs. Awesome, awesome, guys. Okay. For acute gout, the treatment of choice will be non-steroidal anti-inflammatory drugs, and NSAIDs will be used for that. But allopurinol is actually used for the treatment of the chronic gout, or we can say for the maintenance of this one, sir. Next one. Let's go to the next topic right now, guys. Carbohydrates. As I promised you,